coming. This is our second in a series of anthropology lectures called In the Beginning. Dr. Phil Maroney from the physics department started us out on our first one, and he got us through 7 billion years, which was not bad. He took us from the most fraction of a split second after the Big Bang to the creation of the strong and weak nuclear forces, gravity, electromagnetism, and the creation of atoms, gas clouds, suns, galaxies, and we have now had the creation of the Earth some 4.5 billion years ago. So thank you to Dr. Maroney for really getting us off to that much of a beginning. Now we're going to take one more dip into this question about in the beginning. And I'm delighted that Dr. Barry Solinsky from the chemistry department is here to talk about some of the current thinking in chemistry about how, in fact, life began, when it began, and what is life. So thank you so much, Barry.
Thanks. Hi, everybody. Thank you for coming. Thanks for that kind introduction, Lowell. I wanted to begin by just thanking Phil for setting us up for this talk. Phil, about 3 weeks ago, described the beginning of the universe as we know it, beginning with the Big Bang and the Big Bang Theory. And that happened about maybe 10.5, 11 billion years ago. And at that point, matter started out from a very, very small point and expanded out through the universe. And as Lowell mentioned, about 4.5 million years ago, Earth as we know it formed. It formed from piles of rock and elements and matter, which all kind of collected together in this solar system that we're in right now, evolving around the sun that we have as the center of our solar system. And then sometime between that 4.5 billion year point, excuse me, and about 3.8 billion years ago, life began. I'm not going to go into that part of the story today in any detail, but there's pretty good scientific evidence which indicated that life existed on this Earth somewhere between 3.5 and 3.8 billion years ago. So it makes sense that it started at about that time. What I'm going to try to do today is to explain to you a little bit about what life is, what that very primitive life on Earth might have looked like, and how it actually got here. I'm going to keep looking at my notes over here a little bit. So, this is the question. Oh, there we go. This is the question we're going to try to answer today. How was life established on Earth? How did it get here? Whoops. Excuse me a second. Let me just restart this. I'm not sure why it was slow. Try again. There we go. How was life established on Earth? Oh, God. I think there might be time for a few questions. I wanted to begin by putting this up here for a couple of reasons. First, I wanted to give you a little bit about my theological background. I am an evangelical Christian and an old earth creationist, meaning that I do believe that there is a God, and I do believe that God created the entire universe, including life on Earth. However, I'm not going to limit myself by trying to predict how God put life on this Earth. God could have done it in many different ways, including just setting up the conditions for life to start. And so I wanted you to know that because, as you should know by now, every speaker, every writer approaches his subject with a bias. And that bias is going to come through in the talk. And I wanted to warn you what my bias was to begin with so you'll be prepared for it. A lot of today's talk is based on this book, Genesis, by Robert Hazen, who's a very fine writer. And I'd encourage you to take a look at this book sometime. It's really thought-provoking and makes you think a little bit about the origins of life and how things might have started here on Earth. But he writes it from a very anti-religion, anti-Christian perspective. And it comes through very clearly in his writing. So I want to let you know where I was coming from theologically, but I'm not going to let that try to affect me as we try to look at how life began on the Earth. And what I want to do, in order to try to get us started on this, is for us all to try to make life in our own little ways. And what I'm suggesting that we do is we all start out in God School. Here's a picture of God School. It might look a lot like Hogwarts Academy if you've seen any of the Harry Potter pictures. Now suppose you're in high school and you meet with your high school guidance counselor and you take an aptitude test. And the guidance counselor calls you back in and says, you know, you've got a pretty good aptitude for God. I suggest that we try to get you into God school. So they sign up for God school, and they have to get to God school, probably in the same way you get to Hogwarts Academy. There's some train station somewhere where you have to get on track 8.5 and then move through there to get to this esoteric place up in the clouds. And you spend 5.5 years there. I should mention God school is a 6-year program. It's the kind you might expect because you're going to be God at the end, so it's going to take a little bit longer. And you'll meet with all the best teachers there. Moses will be there, Isaiah, maybe Gandhi, maybe a lot of different people, and they'll teach you how to do a bunch of stuff. You'll take a bunch of tests, you know, pop quizzes. What's Nehemiah 12:27? Anybody know? I don't know off the top of my head. You might have to do some class projects, you know, maybe heal some sick people, cure some blind people, that kind of thing. You get through 5.5 years, and then you meet together in this one big classroom, and you start to go through your thesis project. You get in there and your teaching assistant hands you this project, and this will be the first page of that project. Oh, I should mention, it's an obvious assumption that God school is going to be an Augustinian institution, so you'll see this kind of disclaimer, cheating disclaimer as you walk in. But then you'll get your project, and this is your final thesis project in God school. And the thesis project is create life. If you can do that, you can get out. Now there's some conditions associated with getting through God school. You can create life, and you have to use any atomic materials. You can use any atomic materials that are available on the newly formed Earth. These atomic materials will include any elements up to and including the element iron. I'm going to talk a little chemistry tonight, but I'm going to review things as I go, so hopefully that if you have any questions regarding the chemistry, or actually questions at any point, please stop me. But I will take some time to review a little chemistry as we go. Here's your periodic table, which you might remember from way back. And this lists all the elements out that are found on Earth on the basis of their size. And you notice that you have these ones in the top. Hydrogen and helium are really small elements. And then they increase in size until you get to element 26, which is iron. In creating life, you can use any element up to the element iron. So you can use all of these elements over here, but you can't use these elements down here. No gold, no silver. There were no riches to be made on early Earth. But you could use anything up into iron. So in creating life, you can use any element that's available. You can also use small molecules. Small molecules, we're going to define those as any molecules which contain no more than 5 atoms and no more than 3 different atoms. So you can use things like water. Water is an oxygen atom connected to 2 hydrogen atoms. You can use hydrogen cyanide, which is a carbon, a nitrogen, and a hydrogen. Hydrogen cyanide is a very poisonous gas. We'll be talking a lot about that tonight. But you can't use a larger carbon-containing molecule like this one. This is acetic acid. Lots of organisms on Earth can use acetic acid for food these days, but it's unlikely that acetic acid would have been found on that newly formed Earth. These larger carbon-containing molecules would not have been formed at that time. And so in creating life, you have to find some way of making larger molecules like this from smaller molecules like that. Okay? You can also assume any environmental conditions that you need in order to create life. One of the nice things about early Earth is that, as best we know, early Earth had a lot of different environmental conditions available to it. There was all kinds of geothermal activity occurring as the tectonic plates and other geological events happened, moving things around. There'd be lots of volcanic eruptions and other eruptions of molten material from the center of the Earth. It's likely there would be a lot of ice ages, or at least the potential for ice ages on this early Earth. There were still lots of big things hitting Earth at that time, asteroids, meteors, relatively large chunks of stuff, which would cause cataclysmic environmental events. And so if you need some special environmental event in order to get life to occur, you can use it, because chances are almost anything happened during that first 800 million years or so of Earth's life. Now there's good news and bad news associated with this examination. The bad news is there's no partial credit. There's no such thing as a subcellular zombie. Either it's alive or it's not alive. So you have to get to that point. The good news is that you have 800 million years to complete the exam, because it might take that long to do it. Now, as anybody here who has gone through graduate school knows, even though it might be a 6-year program on paper, you don't get out until you finish the thesis. So these guys might be here for a long time before you finally reach that time when you're certified as God. So that's a little bit of a tongue-in-cheek introduction, but that's the kind of thing that origin of life scientists are looking at. What they're trying to do is to figure out what was available on the newly formed Earth, what kind of environmental conditions were there, and how these things might have worked together to generate the molecules and the assemblies that were needed to generate life. And so that's what our task is going to be tonight, to go through and see if we can figure out, at least make some guesses and do some experiments to figure out what kinds of things might have happened and how life might have begun from nothingness. Before we do that, let's make a few assumptions. Let's begin by assuming that all of the organisms on Earth today begin from a single ancestral organism, a single cell or a single piece of life. Now there's some experimental support from this, just observational support. If you look at life as we know it today, all of life as we know it today is pretty similar. Our life is made up of cells, and the cells are separated from their environment by a cellular membrane, a bunch of lipid which separates the contents of the cell from everything else. And all cells that we know of have this sheath of lipid that separate it from their environment. All cells as we know it have genetic material made up of nucleic acids. There's no organism on Earth which does not use nucleic acid as its genetic material. That again is circumstantial evidence to indicate that they all began from the same place, since they all have the same kind of genetic material. They all have similar kinds of metabolism. We'll talk a little bit about metabolism later on, but the pathways that they use to generate energy from molecules and to make larger molecules from smaller ones have common elements, from the simplest bacteria up to the most complicated mammals such as ourselves. So there's a lot of commonality between cells, so it's likely that we all started from the same place. Todd Jackman is giving the next talk, and he's going to talk about evolution, how this process may have occurred. But I have a little pictorial diagram of evolution. I was hoping Todd would be here tonight to enjoy this. But this is just a picture event showing exactly how this happens. We start out with one initial cell, and from this initial cell, everything on Earth evolved. We would generate our dinosaur, cute little fluffy bunnies, and well, maybe we wouldn't get him, but that's all right. There's a corollary to this first assumption. Just because that all life that's currently found on Earth may have come from the same place, this doesn't necessarily mean that that was the only life that ever existed on Earth. Maybe there's things around us that are alive in ways that we don't recognize as life. Maybe our closet door is alive and we just don't understand how it is alive. That kind of thing could happen. Another possibility is that our kind of life, the one with nucleic acid as genetic material, surrounded by a cellular membrane, took over and dominated any other kind of life forms it may have generated. And this is good speculation for science fiction, but without evidence of other life, we can't really look for it. Okay, now what is the simplest kind of life around that we know it? The simplest kind of thing that is around that we recognize as being alive are bacterial cells. And these are a couple of pictures of a bacterial cell. This is a microscopic picture of real bacterial cells. And this is a little cartoon diagram of what a bacterial cell looks like. Now, viruses are things which are sort of alive, but they don't fit the description of really being alive. One of the characteristics of a living organism is the ability to reproduce on its own, to make progeny cells or progeny organisms from an initial parent organism. Viruses can't do that on their own. They require the help of some kind of host cell, something that they will infect, in order for this reproductive process to occur. They are not defined as being alive, but they certainly have many elements common to living things. Anyway, getting back to our bacterial cell is surrounded by this cytoplasmic membrane, as it's called here, a cellular membrane, which defines the cell and separates it from its environment. There's a bunch of nucleic acid in the middle. It's called a nucleoid. In bacterial cells, it's spread out over the entire cell. In our cells, it's contained within an intracellular compartment called the nucleus. This nucleoid contains all the material which this bacterial cell needs to make proteins. All of the information that's needed to make proteins is encoded within that nucleic acid. The proteins themselves are made on these small structures called ribosomes. And there's a molecule called ribonucleic acid, RNA, which takes the genetic message, the protein message from the DNA, from the nucleoid, and takes it over to the ribosome so that protein can be made. We'll talk a little bit more about RNA, ribonucleic acid, at the very end of this talk. There's a whole bunch of semi-solid material in the cell called the cytoplasm. It contains proteins, liquids, small molecule, food, other things. The bacterial cell can move around using this flagella. Some bacterial cells have flagella, some don't. And the bacterial cell has these pili on the outside which maintain communication with the environment. It picks up signals from the environment about what it's touching, what food might be around, and allows the bacterial cell to either move toward those, if they, for example, if it's food, or move away from them if it's dangerous. Bacteria are comprised of a lot of different small molecules and 4 different classes of macromolecules. And we're going to concentrate a lot tonight on looking at 2 classes of macromolecules, nucleic acids and proteins. Nucleic acids and proteins are thought to be very important for life. Certainly without nucleic acids, we cannot have life as we know it. So in order to figure out a way in which we can make living things from relatively small chemicals, we have to go through the step of figuring out how these small chemicals can go and make nucleic acids along the way. Proteins are historically thought to be very important for cells. They may or may not be, as we'll see a little bit later on. Polysaccharides are an important component of today's cells, but they might not have been so important in those primitive cells. And so we're not going to spend much time talking about them. But they are very important in all kinds of cells that we have today. And again, lipids, the material that generates the membranes that surrounds the cell. These are really important, but just for the sake of time, we're not going to talk much about lipids today. That is my research area, lipids and membranes, so it kills me just to let them go without talking about them, but for what we're doing today is not that important. I just want to make a little analogy here. If multicellular organisms are like us, people, we can make the analogy that a multicellular organism is like the King of Prussia Mall. I live about here. I live about a mile from the mall, but I never go there. I'm sure you've all been to the mall. The mall is a big collection of stores, each of which have independent functions. Old Navy sells different stuff than Five Below, than Macy's, than Nordstrom's, than whatever. You get the idea. But there's commonality between them. They share the same utility system. If you buy a general gift card, you can use it at any one of these stores. These things are separate entities which are all closely connected and interrelated and dependent upon one another. That's a multicellular organism like us. A single cellular organism, like a bacterial cell, is like Costco. And Costco is my favorite. My wife is here tonight, and Costco is my favorite store because they have everything and lots of it. If you want to buy soft drinks, you have to buy it by the case, but you can buy Coke, you can buy Pepsi, you can buy Costco brand, you can buy Sprite, you can buy all kinds of stuff. That's the way that cells are today. Bacterial cells are, even though they're the simplest kind of cell that we have today, are really complicated. There are redundant pathways in bacterial cells which allow them to make multiple things. Bacterial cells can eat just about anything. That's why bacteria grow everywhere. You can feed it sugar, you can feed it protein, you can feed it urea. I mean, bacterial cells are really good. They can eat anything, and they can adapt to almost any kind of environment. And the reason they can do that is because there's lots of different things in there, lots of different stuff which allow them to do that. Back in the early days of Earth, back 4.5 billion years ago, cells don't have to be like that. And chances are they're not. The initial cell can't be that complicated. If today's bacterial cells are like Costco, those early cells are like the old general store, where if you went to get a loaf of bread, you would pick up one of the 2 or 3 loaves of bread on the shelf and take it home. There wouldn't be a choice between rye and wheat and whole wheat and multigrain and 6 grain and 12 grain. There'd be one kind of bread there. There'd be one kind of sugar. And that's the kind of thing that we have to keep in mind as we look for that primitive, basic organism, that it's going to be really simple, and we need to take a real minimalist approach at looking at the types of stuff that's necessary for this cell to exist and for it to be alive. To be alive. What does it mean to be alive? Well, there was a book published a few years ago called Biogenesis. And the author of Biogenesis, a guy named Lahav, took statements from 48 different origin of life scientists, researchers who are interested in learning about how life began. And he went to them and asked them, what is life? How do you define what life is? And he was very surprised to get 48 definitions which were not that similar to each other. There was a lot of difference between them. I'll just put a few up here. John Maynard Smith says that life is any population of entities which has the properties of multiplication, heredity, and variation. Multiplication means that it can divide. If you have a single-cell organism, it can divide into many daughter progeny organisms which can go on and divide and conquer. Heredity. It can pass genetic information from one generation down to the next. And variation. Not all of these single-cell organisms are the same. There are differences in their genetic information. That's great, but it doesn't say anything about what they do, where they live, how they eat. So there's some limits to that kind of definition. Another definition, Stuart Kaufman. Life is an expected, collectively self-organized property of catalytic polymers. Catalytic polymers would include proteins and nucleic acids, what we talked about previously. In this case, it talks about what the cell might be, what the life might be. It's a bunch of stuff put together. But it doesn't say anything about what the life does, nor does it say anything about the transfer of genetic material, heredity, from one generation to the next. Norman Horowitz, life possesses the properties of replication, catalysis, and mutability. He's now talking about catalysis, the ability that life has to catalyze chemical reactions, to take big molecules and to break them down chemically to make food and energy out of them, or to take little molecules and build them up to make bigger molecules out of them, make something that the cell, that the living organism can use in order to improve itself, to build its cell up, to strengthen itself, to carry out other chemical reactions. The definition that Hazen likes, and the one that I'm going to spend a little bit of time talking about, is this one, which was developed by a microbiologist named Gerald Joyce. Life is a self-sustained chemical system capable of undergoing Darwinian evolution. This definition kind of gives you some description on what you might expect from that early living organism. And in particular, it describes some minimal elements that this initial life must have had to have. The first thing that statement says that life must be chemical. One of the things that many computer scientists are doing these days is to try to generate artificial intelligence, computers that have the ability to think, to learn, and to make decisions without our input. So anything computer generated that we're working on right now is not defined as life. It has to be chemical. It has to involve molecules like the ones that we saw early on in the slide and the making and breaking of these molecules. It can't just be a bunch of electrons moving back and forth within a bunch of semiconductors. Life must sustain itself by gathering atoms and energies from its environment. It's got to eat. If it's alive, it has to eat, and it requires metabolism. It requires those reactions which will make things and break things. Living organisms must display variation. One characteristic about all of us in the room is that we're all different from each other. You look around, you don't find any clones of yourself in here. This is true of every kind of organism. I'm working with an organism, I'm on sabbatical leave right now, and I'm working with a bacteria called Escherichia coli. It's a standard experimental bacteria that everybody uses. There are thousands of strains of Escherichia coli. So even though these organisms are very consistent and very, very similar to each other, there's still genetic variability between them. And that is something that is characteristic of life. Life is not a bunch of cloned, identical entities, exactly the same. There's variability involved which allows us to adapt to our environment, which allows us to evolve, if you will, if you like that word. So let's get to the nitty-gritty a little bit. If we're on this very early Earth and we want to make life, the first thing we have to figure out how to do is to take those chemicals, small molecules that are available to us, elements, and make them into something that can be used for life. The two things that I'm going to concentrate on are generation of nucleic acids and the generation of proteins. Both of these things are biopolymers, biomacromolecules. A polymer is a large molecule which is generated, which is built from connecting together a series of related small molecules. Nucleic acids are, in particular, polymers of nucleotides. And I'll show you the structure of a nucleotide a little bit later on. So if we start out on primordial Earth, pre-biotic Earth, with very small molecules and elements, we have to define a way, or figure out a way, or figure out how on this early Earth these small molecules and elements were converted into these nucleotides necessary to make the polymers, nucleic acids. Excuse me. Same thing happened with proteins. Proteins are polymers of a different kind of building block, a different kind of a building material called amino acids. Again, amino acids, they're not big, but relative to those small molecules and elements, they are kind of big. And so we have to look and see how possibly those elements and small molecules on early Earth were made into amino acids, which then could be made into proteins. So how can we make these? Origin of life scientists have been thinking about this for a fair amount of time. Research really started in earnest. Some of the most famous experiments were done by a man named Stanley Miller when he was a graduate student in the lab of Uri, U-R-R-Y. I forget the guy's first name. I apologize for that.
Harold.
Harold, thank you. Thank you very much. And what Miller did was he tried to simulate the environment on early Earth and to see if, in this environment, if the small molecules that were available at the time could be made into amino acids, the components of protein. In the 1950s, it was still believed that proteins were the center of the universe, of the living universe, that is. It was about this time when the double helical structure of DNA was figured out by Watson and Crick, but people still didn't appreciate the importance of nucleic acids in the cell. They still thought that most cellular things happened because of protein. And Miller focused on how these proteins and the amino acids that made up these proteins, how these would have been generated on very early Earth. And so this is a little bit of a cartoon summary of the experiments that Miller carried out. Miller generated this type of apparatus which included a big bunch of boiling water down here. Early Earth would have been largely covered with oceans, and early Earth would have been very warm. So you would have had an atmosphere which was saturated with water vapor, and Miller simulated that by generating this heated compartment filled with water. He had an evacuated chamber up here, which he filled with some of the things that people thought would have been prevalent in the atmosphere on early Earth. Early Earth's atmosphere included this molecule, ammonia. If you've ever opened up a bottle of household ammonia, you know how nasty it smells. It was thought there was a fair amount of ammonia in our atmosphere. Hydrogen gas. Hydrogen gas was the stuff that they used to fill up dirigibles on. It's the stuff that blew up when the Hindenburg blew up in the 1930s. Highly flammable gas. Methane is the major component of natural gas, another highly flammable gas. Carbon dioxide, which we all know about from greenhouse gases and global warming, can't talk today, and the water vapor coming up. So Miller took this chamber, filled them up with these gases, and then hit these gases with an electrical discharge to try to simulate the types of lightning storms, electrical storms, which were thought to be prevalent on early Earth. He did this for a couple of days and then saw what developed. And after waiting a few days, he saw this brown goo develop, which worked its way down into this water compartment down here. And he analyzed this brown goo to see if anything interesting came out of it. This was one of the experiments that he did. He mixed together methane, ammonia, and hydrogen gas at a ratio of 2 to 2 to 1, heated these up for a couple of days with electrical discharge, and saw what came out. What he saw was a fair amount of this molecule, which is glycine. Glycine is one of the amino acids that's found in the proteins in our cells. So this was very exciting. What he proved is that on very early Earth, using simulated environmental conditions which were consistent with what we knew about early Earth, you could take the small molecules that were available and make them into things which subsequently could be made into biomolecules, made into proteins. He changed the composition of his flask and found he could make most of the amino acids that are found in today's proteins. So this was pretty exciting. What he found was that you could make the components of these proteins, of these long polymers, by simple chemical means, making reasonable assumptions about the environment at that time. A little bit later, another researcher named Oro carried out this experiment where he took hydrogen cyanide. Again, hydrogen cyanide is a very poisonous gas, but again, it was believed that hydrogen cyanide concentrations were fairly high in the atmosphere of early Earth. And he just heated these things up, and he found that heated hydrogen cyanide would spontaneously form this compound, which is the compound adenine. Adenine is part of the nucleotides that are necessary to make nucleic acids. So this, again, was another exciting discovery that one could take a poisonous small molecule and just by simply heating it up, make something that could be made into a biomacromolecule. A lot of different people, without getting into details, went through and did a lot of parallel experiments to demonstrate that most of the small molecules that were needed to make these biomacromolecules could be made by just varying environmental conditions, heating, cooling, electrical discharges, the composition of the gases that were mixed together, and you could make most, if not all, the components necessary to make the biomacromolecules you need for life. So we've made the building blocks experimentally. The next step is to take these building blocks and then make them into long biopolymers, biomacromolecules, which are needed for life. The small molecules are great, but they're not alive. In order to get life, we need to make these bigger things. How do you get the smaller things connected into bigger things? Well, the big problem associated with this assembly of biopolymers is that even if you have the generation of amino acids and nucleotides through these gaseous reactions, these would get down into the early oceans and generate fairly dilute solutions of these things in early oceans. If you need, say, for example, 2 amino acids to get together to form the first part of a protein, they've got to be close to each other to do it. They've got to physically bump into each other. And at the concentrations that they would be found in this early Earth, it's unlikely these guys would just find themselves and bump into each other with the right orientation and sufficient energy in order to get a reaction to get them to stick to each other. And so that's a problem that needed to be overcome. How possibly could we get these amino acids or nucleotides close enough to each other to get them to stick together, to get them to connect? Well, one possibility is to have these nucleotides or amino acids assemble themselves onto crystalline templates, which would have been all over the place on early Earth. Let me put forth this scenario for you. Suppose you have an ocean and a seashore. And the seashore, the ocean is splashing up against this rocky shore, which would have an indentation in it. So the ocean would splash in, a bunch of seawater would splash up into this indentation and create something akin to a tidal pool. At the bottom of this indentation would be this crystalline material, some kind of inorganic template with a bunch of regular imperfections in it, very small at the molecular level, into which, say, an amino acid could settle. So we have a dilute solution of amino acids splash up into this rock. The amino acid would be in this solution. Early Earth would be fairly hot. The indentation would heat up either from the heat of the sun or perhaps from some geothermal heat, and the water would boil off. And the amino acids would concentrate down as the water boiled off and eventually find their way into a couple of these little indentations in this crystalline lattice. Another wave would come in with a few more amino acids, and they would find their way into these indentations. And this would happen over and over again for as long as it takes. Again, it took 800 million years or so between the beginning of Earth and when life started. So this process could take thousands and thousands of years, perhaps. If these things get close enough together, between their close proximity and the heat of the sun, it's very reasonable that these things would get in here, line up next to each other, and form the chemical connections that are needed for the biopolymers. So this is one reasonable hypothesis that one can propose, which would allow the formation of biomacromolecules beginning with a dilute solution of the building blocks necessary to do it. I'm not saying this is the way that it happened, but it's at least a reasonable suggestion, a reasonable theory which one could use to explain the production of these biomacromolecules. So, you've gotten through this now. You're working with your friends in God School on this project. You figured out a way to make the building blocks for the biopolymers. You've got a reasonable hypothesis for getting these biopolymers made from the building blocks. You write this stuff up, you hand it into your teaching assistant to have a look at, and the teaching assistant comes back to you and says, you know, you've overlooked the chirality issue. And you say, well, what's that? Chirality is a property associated with some molecules, including all of the molecules that are needed to make these biopolymers. The best example of chirality are your hands. You have two hands. Each hand is a palm, a thumb, and 4 fingers. Both hands have a pinky, both hands have a ring finger, so on. The definition for molecules to be identical is that they have to be superimposable. You have to be able to take one and squash it on top of the other and have it look exactly the same. Your hands are essentially identical. They do the same things. They have the same components. But if you try to superimpose them, they don't superimpose. Put the fingers on top of each other, the fingers don't line up right, and the thumbs are in the wrong position. The hands are not perfectly identical. They're mirror images of each other. If you had a mirror over here and held your hand up, you would see a reflection which would look a lot like your left hand, but they're not identical. The issue is that the building blocks that are used to make these biopolymers have the property of chirality. These are kind of cartoon representations of amino acids, the building blocks that are needed to make up proteins. Nucleotides have sugars on them that work in the same way. A little easier to see from amino acids. Amino acids have common structures. They have this central carbon atom. They have an ammonia group, an NH3, sticking off from one side. They have a hydrogen atom sticking off on the other side, a carboxylic acid, and then something else. And the something else could be one of 20 different things, and that's what makes 20 different amino acids in our proteins. If you look at how these 4 different things can be arranged around the center of this molecule, you can find that they can be arranged in 2 different ways. If you put the carboxylic acid part, the green part on the top, and the red part on the bottom, in 2 different ways, you can either have the ammonia group, the blue part, to the left, and the hydrogen to the right, or the hydrogen to the left and the ammonia group to the right. These 2 things are not superimposable. You can't lay them on top of each other and have it be the same. They're mirror images of each other. If you had a mirror here, this thing would be the mirror image of that, and vice versa. The problem is, and this is the chirality issue, if you link these things together, if you connect these to make a long biopolymer, the biopolymer you make when you use L amino acids, one form, is different from the biopolymer you make when you use D amino acids. They're shaped differently. The way that the connection would come off in a different direction. If an L amino acid would come off of a central part like me, straight in front of you, a D amino acid may come off down here somewhere. So the 3-dimensional shape of these biomacromolecules is extremely important. If they don't have the right shape, they don't have the right function. And so you can't arbitrarily use one form or both forms of these amino acids. You have to choose one in order to get a reproducible 3-dimensional shape for your biomacromolecule. So, as our TA in God School correctly points out, in designing these biomacromolecules, if we're going to try to design the ones that we are currently aware of, we have to take this into account. We have to figure out some way in which, using our environmental forces and the materials that are available to us at the time, that we can select for one form over another. And that's a problem that people are working on right now. There are certain kinds of inorganic materials, inorganic clays and crystalline materials, which themselves have a orderedness to it which allows them to prefer one form of amino acid to another. And some very careful studies have been shown to indicate that these kind of crystal materials will select for, say, this form of an amino acid over that form. The problem is that the preference is really, really small. It's only 1 or 2%. And so from the information we have right now, we don't know how those early clays might have selected for one form over another. It could be the early clays had a crystalline structure so very different from what we're aware of now that they might have been able to distinguish between the 2. but we don't have any good experimental evidence for how this kind of selection process might have occurred. So that's something that's going to keep these potential gods in God school for some time as they figure this out. Another problem that our TA pointed out to these students is the entropy problem. Our universe can be described by a number of different physical laws, which describes a lot of the energetic behavior, the physical behavior experienced in this universe. One set of laws we describe this are the laws of thermodynamics. And the law which we're interested in here is the second law of thermodynamics, a law which describes a quantity called entropy. The second law of thermodynamics states that in any spontaneous process, there is always an increase in the entropy of the universe. What that means is that given the opportunity, any ordered system is going to strive to become more disordered. Suppose you're in your house and you're up in your bedroom upstairs. Some of you guys probably remember that. And your mom's baking cookies downstairs. And you're not aware that your mom's baking cookies until you smell them. The reason that you smell the cookies is because of entropy. Mom is baking cookies, and these cookies are in this warm oven, and heating up the cookie dough allows it to give off these wonderful smells. Now, without entropy, these wonderful smells might stay right next to the cookies. They would just hang out there. They'd be nicely ordered all the way around the cookies and just kind of get stuck in the oven and not get out. But entropy says, oh no, these things want to get all over everywhere. So they escape from the oven, they escape into the kitchen, they escape into the living room, they go upstairs, they escape into your room, and they go all over Earth until they finally become so diffuse that you can't smell them anymore. That's what entropy says. If I had a bucket of marbles and dropped them on the floor, the bucket of marbles wouldn't roll over in the corner and stay there. They would spread out all over the room as best they could. In order to make life is very ordered. We are very ordered things. And in order for us to be ordered, we defy the second law of thermodynamics. The reason that we defy it is because we generate a lot of energy. And we use the energy that we generate to overcome that. But on early Earth, we don't have the ability, life doesn't have the ability to generate that energy before the fact. Life has to form first in the absence of this energy that holds us together. And so one has to explain that a little bit. One way in which that can be explained is by a theory of emergence, that there is some force that we don't understand yet, which helps to provide order to things in a universe that would prefer chaos. Another way to explain that is again, there's energy available to us in the universe, little bits of energy, which we're exposed to all the time. And this little bit of energy could drive systems from systems of more disorder into systems of more order. And Hazen in his book uses a few natural examples of this, one of which I show up here, birds flocking together. There are lots of good reasons for birds to flock together. It helps to protect them. It helps them to find suitable mates. I don't know, it's nice to go in a group. I don't know exactly what other things might work. But the issue is that even though there's lots of good reasons for them to fly in a flock, there's no physical force holding them together. They're not holding wings as they fly through the earth. There is some other force which is driving them to flock. Some innate understanding, some innate desire for them to flock together. Schools, or fish move around in schools. Clouds get together. And the reason that is there's some kind of energetic force which is driving these things to become ordered when there are other forces pushing them apart. We don't have a really good understanding yet on how these kind of things happen. We can observe some natural effects which suggest that they happen. And so hopefully or possibly there will be some better understanding of this emergence theory, this ordering effect as time goes on. But again, it's something that our God School participants have to take into account as they try to generate life, because it's important that this ordering effect take place. It's absolutely necessary in order to push the things together, the components together that are necessary to generate a living organism. Okay, for the last couple of minutes, I don't want to run too late. I want to talk about the two most likely explanations for how life might have begun on this planet. There have been a number of different hypotheses on how life might have begun, some better than others. There was one called a protenoid hypothesis, where if you took a bunch of amino acids and put them on a hot rock, they would get together and form this mass of gooey material, which was termed a proteinoid. And the proteinoid kind of looked like life, it kind of looked like a protein, and kind of didn't. Some people liked the idea for a while until the guy who discovered the proteinoids decided that these things had a life of their own and an innate consciousness, and thought a little bit. And about that point, some of the other scientists in this area felt this guy might have been a few cards short of a deck and had him move on. So there have been lots of different hypotheses, some of which have been pretty good, some of which are not so good. But there are two that really stand out right now as being well-supported and legitimate hypotheses which might explain the beginning of life from non-living stuff. And the first one is this iron-sulfur world. The iron-sulfur world hypothesis was generated by, I don't want to mess the name up, Gunther Wachthuizen in about 1988 or so. And what he suggested was that life began at hydrothermal vents at the bottom of the ocean. Up until this point, people like Stanley Miller and most of the other original life scientists believed that life began at the interface between water and land up on the top of the Earth. The reason it would begin there is because it had lots of environmental pressures associated with it. There'd be lots of heat, there'd be sunlight, there'd be lots of water, there'd be the ability to interact with clays and rocks, there'd be the atmosphere, which would be a natural source of starting materials. There's a lot of good reasons for life to begin at that point. What Wachthuizen suggested was that life actually begins at these hydrothermal vents. This is a picture of a hydrothermal vent. You got this big kind of tower here, and you have gases spewing out of the towers. The gases that are spewing out of the towers are volcanic gas containing largely a gas called hydrogen sulfide. This plume is also going to contain a lot of steam, a lot of boiling water, because the hydrogen sulfide and other volcanic gases that are coming up from this Earth are going to be really, really hot relative to water, which boils at 100 degrees Celsius. So what Wachthuizen suggests is that the hydrogen sulfide and other gases that are coming out of these geothermal vents provide a natural food source and also a natural energy source which could be used to fuel the reactions that could lead to the reactions generated by living organisms. Let me talk a little bit of chemistry. Again, one of the elements prevalent on early Earth would be iron. There's lots of iron in the rocks and oceans and all over the place on early Earth. Iron will react with hydrogen sulfide in a couple of steps to form first this molecule, which is iron sulfide and hydrogen gas, and this reaction generates a fair amount of energy. The iron sulfide can react with a second molecule of hydrogen sulfide. Hydrogen sulfide is kind of a stinky stuff, by the way. If you ever smell that rotten egg smell from burning tires or that type of thing, a lot of that is hydrogen sulfide. Hydrogen sulfide will react with a second molecule of iron sulfide. Get that right? Iron sulfide will react with hydrogen sulfide to generate this material, which is a mineral called pyrite, which is really prevalent around these hydrothermal vents, and more hydrogen gas and energy. So initially, you get these chemical reactions occurring, which would generate a fair amount of energy, which could be used to fuel reactions necessary for the generation of life. Now, what Wachthuizen suggests is that around these geothermal vents, a lot of reactions occur which would have generated carbon-based molecules which would be necessary for life, and then one could generate a chemically based metabolic pathway similar to that found in our cells. The pathway that's suggested by Wachthuizen, and this one is reverse citric acid cycle. This is found in a few primitive organisms that are located, that have been isolated from these hydrothermal vents or on the edges of these hydrothermal vents. The way that this reaction works, if you've had biology, I don't know how many biology students are here, but one of the pathways that generates a great deal of energy in our cells is called the citric acid cycle. We will eat sugars, and these sugars will be broken down into a molecule called acetyl coenzyme A. Acetyl coenzyme A will react in the citric acid cycle with oxaloacetate to make a molecule citrate. Citrate will get broken down in a number of steps to this molecule, succinyl coenzyme A, and 2 molecules of carbon dioxide will be given off in this breakdown process. But the other thing that happens in this degradative process is the generation of a bunch of energy. And our cells have the ability to capture this energy, the energy that's generated in the emission of this carbon dioxide and what is the equivalent of hydrogen gas. Our cells have the ability to collect this energy and save it in the form of adenosine triphosphate, ATP, which is the chemical storage molecule found in all of our cells. Most organisms have this citric acid cycle, or some very close form of the citric acid cycle, and they use it to generate energy from carbon-based material that these cells will consume. Now, a few primitive cells around these geothermal vents have a reverse citric acid cycle, where you begin with acetyl coenzyme A, and you add, in a stepwise fashion, a number of different gases to it, gradually increasing the size of this molecule until you get back to citrate. I guess I should mention, for the people who aren't too familiar with this, acetyl coenzyme A has 2 carbons in it, along with a coenzyme A, which just holds those 2 carbons in place. In the reverse citric acid cycle, we add a molecule of carbon dioxide to generate a 3-carbon molecule, pyruvate. Add a second one to generate a 4-carbon molecule, oxaloacetate. Add a molecule of hydrogen, a molecule of water, another molecule of hydrogen, another molecule of carbon dioxide to generate a 5-carbon molecule, this 2-oxoglutarate, and then finally one more molecule of carbon dioxide, here it is, to generate isocitrate, which in a couple of steps is made back into citrate. So we're starting with molecules with virtually no carbons, 2 carbons as in acetyl coenzyme A, and making molecules with 6 carbons. This 6-carbon molecule can break down into a 4-carbon molecule and the initial 2-carbon molecule. And this 4-carbon oxaloacetate, I probably should have put one more arrow here, can go on and make other stuff. It can carry out other reactions which will make it into bigger and bigger things. One of the things that our cells do is they can take oxaloacetate and convert it into amino acids. The amino acid aspartic acid, for example, is generated from oxaloacetate in one step. So this is a reasonable set of chemical reactions which might be used to generate larger carbon molecules from smaller carbon molecules. A couple of nice things about this. One is that all of these gases that are used to add on to this initial acetyl coenzyme A are readily available in seawater around these hydrothermal vents. Lots of carbon dioxide is available. Lots of hydrogen gas is generated due to that breakdown of hydrogen sulfide. And there's lots of water around, of course. We're sitting in an ocean. Another attractive thing is that Wagerschausser suggests that these reactions can be catalyzed by complexes of iron and sulfur that would have been located in large amounts around these hydrothermal vents. We have hydrogen sulfide reacting with iron, generating pyrite. Pyrite is a combination of iron and sulfur. A curious, or maybe not so curious, coincidence is that if you look at these primitive organisms that have this reverse citric acid cycle, these reactions in those organisms are catalyzed by proteins. But the proteins contain an iron-sulfur complex within them, which is necessary to catalyze these reactions. So it's not a huge step to think about a series of truly chemical reactions requiring an iron-sulfur complex, and then just by a natural evolution, having these iron-sulfur complexes, these catalysts, contained within protein molecules, which somehow find their way to this site. And so this is a natural generation of metabolism, which by most definitions of life is required for life. So it's a small step from a bunch of chemical reactions occurring to a bunch of chemical reactions within a contained, controlled system. And that's pretty much what that slide says. Now there's one problem with the iron-sulfur hypothesis, is that that really doesn't tell you how genetics occur. It really gives a nice demonstration on how metabolism started, how cells might have conducted the chemical reactions needed for life, but it doesn't talk about how genetic material is generated, how this metabolic pathway might be transferred from one cell to another cell to another cell, where reproduction takes place. So that's the one criticism that one might have of this iron-sulfur world. A different hypothesis, the second hypothesis I wanted to mention, is the RNA world. And there's a book on this called The RNA World, which you might choose to look up. And it describes in great detail how life might have begun simply by ribonucleic acid molecules. In the RNA world, the hypothesis is that life began as a collection of a small number of self-reproducing catalytic RNA molecules located within a small area. Again, up until about 25 years ago, people didn't think much of RNA. They thought RNA was kind of boring. It was a messenger molecule that carried information from DNA, the nucleic acid genetic material within our cells, to ribosomes in order to have them make proteins. But about 25 years ago, oh, I should mention what RNA is for people who aren't really up on that. RNA is a polymer of nucleic acids, and it kind of looks like this chemically. It has this base structure, which includes adenine, guanine, cytosine, and uracil. One of the early slides that I put up had a picture of adenine on it, so you know kind of what adenine looks like. These three look not that different from sugar, a ribose sugar, which is sitting here, D-ribose to be precise, another one of these chiral molecules. And these ribose sugars are connected together by these linkers, which are phosphate groups. This is a three-dimensional RNA molecule. This type of molecule is called a transfer RNA molecule. It's necessary in making proteins. One end of this molecule hooks up with another RNA molecule, messenger RNA, and this end of the molecule has an amino acid stuck on it. So this thing acts as an interpreter, interpreting a DNA sequence and making it into a protein. This is what kind of gives you a three-dimensional representation of what an RNA molecule looks like. So again, up until about 25 years ago, RNA was boring. But about that time, Tom Cech and Sidney Altman discovered catalytic RNA. They discovered that some RNA molecules had the ability to catalyze chemical reactions. Up until that time, people thought that only proteins had the ability, in terms of biological molecules, to catalyze chemical reactions. Now, in terms of origin of life research, this was a huge discovery. Up until that time, people thought we needed proteins and nucleic acid within our cell to generate something that was alive. If we know that nucleic acid can be genetic material, if nucleic acid can also be catalytic material, we now only need one molecule to do everything. And that makes life a lot simpler. We don't have to make 3 or 4 different kinds of molecules and get them together. We only need to make one kind of molecule. A little bit later, in the mid-1990s, it was discovered that some RNA molecules are self-replicating, that they can make exact copies of themselves if they just have some of the building blocks, the ribonucleic acids around to do that. This is another huge discovery. For our cells to divide, what has to happen is that we have double-stranded DNA in the center of our cells, and a whole series of proteins and nucleic acids work on this double-stranded DNA to get cell division to occur. There's one protein that separates the double-stranded DNA. There's another one that sticks in there to keep it from getting back together. There's another series of proteins that copy the one strand of DNA relative to another. There are more proteins that push the new double-stranded DNA apart from each other. There's a whole series of things that have to happen for cell division to take place. If one has an RNA molecule which can make a copy of itself, be its own Xerox machine, again, you've greatly simplified the problem. You don't have to include all these other elements if you're trying to define what early life looks like. We can have an RNA molecule which can be catalytic, which can catalyze chemical reactions, but which can also replicate itself. So these types of molecules would have both metabolic capability and genetic variability associated with it, which, again, greatly simplifies what that early life, what that first cell might have looked like. There are some problems associated with this. We have not yet been able to experimentally demonstrate large replicating RNA molecules. They can only replicate themselves, the one that has been designed to this point, up to about 14 nucleotides long. In order for an RNA molecule to be catalytic, they need to be somewhat bigger than that. The other problem associated with this model is the statistical problem. You need to have tens of these things get together in order to do enough catalysis to generate reasonable metabolism. And it's statistically difficult for 10 or 20 of these distinct RNA molecules to get together and work cooperatively. but they had 800 million years to do it, so a lot of time can overcome a lot of statistical unfavorability. I am kind of fixated on zombies, so I just thought I'd throw a zombie slide up. And I'd like to thank you for coming, and I'd be very happy to answer any questions you might have. Sugar.
You said, so it's actually a combination, you said.
Right.
Well, humans, well, animals can only absorb L amino acids. And if they go into a structure in the body like the teeth where they don't change, then they rationalize. They go back towards a 50-50 split. So if you die and somebody looks at your skeleton 100 years from now, you're going to find a mixture of D and L amino acids. So only the L amino acids come in the first place.
That's one of the ways that you can date. It's not common to use to date stuff, but you can use that to date how old something is. It's only good for, like you say, a few hundred years, so it's like that, used that often. Yeah, Phil.
Harry, I was wondering about the entropy thing. Again, time may be able to overcome that in long enough time. For instance, take the air molecules in this room. You took a snapshot of it. The probability of that happening again just that way is incredibly small. And every combination of velocities is equally likely. Any one to reproduce is very difficult. And that's also for the distribution of molecules all going that way. That's equally likely. But when that happens, see, when all the other ones happen, it's indistinguishable. We don't see any change in the room. When all the molecules decide to go that way, something very dramatic happens, and it looks very ordered. But it has the same probability as any other configuration. But given enough time, it'll blow out the wall. We won't know what, but it'll look like a very ordered event. I'm just wondering what kind of probabilities are involved to have this entropy look ordered to start this process. How much time would be involved using the probability and the size of the sample and things like that. I just wondered about that.
Yeah, I have some colleagues I think that would love to answer that question, or would love to think about that question. That's the kind of thing I don't normally think about. I really don't know. There are certainly lots of forces around that order things. That
Well, this doesn't require a new force. It just requires waiting long enough for something that looks disordered to look ordered.
right. There's a time factor. How long will
How long does it
be
take?
going to maintain order too, in order to allow that to happen?
Right.
Will the other entities be present at that instant in time when it becomes ordered?
Given enough time, basically anything can happen. One of my favorite examples is that if you were to turn and walk into this wall over here, given enough time, all the molecules in your body would align with all the spaces between the molecules and the wall, and you'd walk right through the wall. But it would take an enormously long period of time.
Right, we can do that calculation. Right. We talked about that.
It's just a question of figuring out how many molecules there are here and there.
It's several times the age of the universe.
I'm sure it is.
So, that's what I was wondering if anybody had done a calculation like that.
Yeah, I'm not aware of that.
The probability says it may happen right now, so we're trying.
You want to start trying? We've got time.
Yes.
Is there any evidence for life as opposed to starting here on Earth, arriving here either through an asteroid or some other far from equilibrium?
Asteroids, there's been analysis of asteroids coming in, and asteroids do contain a lot of these biomolecules. They contain nucleic acids, for example. And there was a study in Science, I believe it was published in Science this year, which found these nucleotides on asteroids and developed a reasonable hypothesis for how they got there. They go through a different kind of process than the one that I showed on here, but it's a deep space process, so it has to happen in cold for one thing, and it has to have the materials available on the surface of the, I think it's an asteroid or meteor, I forget which, but yes, these things could have been developed in space and then landed on Earth, and that's one of the things that, one of the hypotheses has been generated. That works for small molecules that's been demonstrated, but not really big things. So that's an issue that had to be overcome.
There's no other question.
Which hypothesis do you favor of these various things that you present?
I'm a creationist, so I don't know. I have some weird ideas about how life got here and about how people got here, and I don't want to share those because they're just too weird. The RNA, I mean, of these hypotheses, the RNA world makes the most sense because it requires the least amount of stuff. It requires a single kind of molecule. And so if the conditions were made so you could make nucleotides, they, you know, in some little part of an ocean somewhere, one can imagine the assembly of thousands of different RNA molecules at one point, which the right combination of 20 or 30 would generate something which would seem alive. So if these hypotheses, that one makes the most sense. Or it could be a combination of, one could have a bunch of RNA molecules around a geothermal vent and they had coffee together one day and decided it would be a good idea to work together. So that, but the RNA one seems to be the best one.
But you haven't gotten to your area of expertise, and that's the lipids to get the
Right.
shell around the chemicals that want to do the research.
That's right. I didn't do that because I'm not sure that's necessary. If you had the right hole in a rock where a bunch of these things could get into and not get out of, the soap bubble or the lipid to make the membrane might come along later and kind of drag it out of there. So it's, in terms of the chicken and the egg argument, I think with respect to life, life requires metabolism and genetic material. And the container for that could have been nothing or something nondescript. One of the hypotheses pushed by Hazen in this book is the flat world, where you had material depositing on rocks, just say the proteins and nucleic acids that are necessary for life. And these things would just stay there because there'd be no outside force trying to attack it. And then at some point, this material would be chopped up into smaller pieces and encapsulated. So that's a reasonable, as good a suggestion as any, as good as argument as any. So I think membranes are after the fact. Yeah.
Can I push you a little bit on your crazy ideas? Because over here on the humanities side of the house, of course, we're fascinated by the idea that somehow we get from what you're talking about here, this gradual development of more ordered and more complex, emergent, something that leads to life, to an institution which insists that there's ideas of God that are important. So is God external to this process, or is it what we call this process of emergent order leading to life? Is it internal? How do you put your two lives together, the creationist life with a life of evidence?
Right. Well, let me preface that. I had a conversation with a colleague of mine in chemistry who's also a Christian, and we think exactly the same way on this, is that these are details. And God is a creator, and how God created is up to him. You know? And for me to speculate and guess on it is just speculation. And hopefully I will get to heaven someday and God will explain it all to me and I'll look around and say, that was pretty obvious from the evidence on there. I should have figured that out. But that said, personally, I don't know how this began. It could have begun with God sticking a pin in that little pile of stuff at the beginning of the Big Bang and then letting it go from there. Or he could have dropped the first cell down on Earth. Francis Crick, who was one of Watson and Crick, who generated the hypothesis about the DNA double helix. This was back in the 60s, so there wasn't a lot of research done at that point. But he did a lot of thought about how life began on Earth. And his conclusion was that there was no possible way that life could have begun on Earth from simple chemicals. So that aliens came down and dropped life here, and that's how life got started. He was totally averse to the idea of a God, and so even though he could not believe that life began from simple chemicals, he was not willing to think that a God actually put life here. So I'm not sure whether God dropped a cell here or God put together the conditions to allow a cell to be created. And I'm happy with either one of those. Where I think that, I mean, I'm not even sure I talked to the wife about this, but where I think is God is important is in people. In that the step from, I'm perfectly happy with evolution from a single cell organism all the way up to apes. But we have a consciousness. We have a soul, if you will, which I don't think any other thing has. And the evolution of a soul from an ape to a human, I just can't understand. I just don't understand the biological reason for us to think the way we do. Why are we compassionate? Why do we care about things? Why do we feel guilty? Why do we have so many emotions which don't make any sense in terms of the survival of the species? And my only explanation for that is that sometime in this point where man or ape became man, God came down and intervened and said, okay, this is how you're going to be special, and gave us these characteristics, gave us the ability to think and to feel and to rationalize. I have no evidence for that. I don't think anybody could, but that's how I rationalize God and this stuff. I mean, for people in here who are religious or Christians, God is an experience. God is involved in our everyday lives in special ways. And it's totally above and beyond biology. So that's the way I think. So, yeah.
Does your model allow for the probability of life existing on other worlds elsewhere in the cosmos?
My personal theology or the chemistry?
Well, the chemistry, obviously. But
obviously, if there are other worlds with similar conditions to this one, life could certainly begin in the same manner. It seems likely that there would be worlds with similar conditions to this. There are growing evidence of planets with atmospheres out in the world, and especially in the last 5 or 6 years. Phil would be more aware of that than I am. And it seems that, I haven't heard much lately, but there were periods where every month they would come up and talk about a new potential planet elsewhere. So there's no reason not to believe that life would be on these planets if it began in this fashion. Because if we all began from the same origin, these planets are going to contain the same collection of elements, same collection of small molecules. The only thing that's going to change is the environmental conditions, how far they are from the sun, what type of stuff might be contained within them. Does that sound reasonable, Phil? Is that?
Yeah, actually, you're absolutely correct from what I understand it. The only thing I would add to that would be that the place of Earth in our own solar system is so precarious with position to the sun, as you mentioned. A little bit in further, a little bit out further. Life may have started on Mars, but it couldn't sustain it because of the atmosphere. It didn't last long enough. We're in our 3rd generation atmosphere. And we had a long enough time at this temperature and very hospitable environment to evolve life, whereas Venus and Mars didn't have that long. Venus was different in the beginning, they had different atmospheres. So, yeah, there may be many stars out there, billions and billions of stars with planets around them, but man, the conditions for life are not as easy as some people think they are. And that's my personal opinion. When you just look at our solar system, a little bit in, a little bit out, there may have been life, but not long enough for it to evolve like into intelligent life like we are. It takes quite a long time. And if your planet's not at the right spot for a long enough time, you're not going to have it. And we only have another 4.5 billion years to get smarter. And then we're going to get all sun, so.
Another question? Did I see a hand? Yeah.
Yeah. Has there been any speculation on other types of life? I mean, everything we know is based on molecules that pretty much involve carbon and the RNA and DNA. Is there any speculation that there will be some type of life that was built up with a different chemical structure than ours that wouldn't, that was completely different? Or is that just out of the question?
There's tons of science fiction on it, and I'm a big science fiction fan. And there are things like Andromeda Strain with silicon-based life. And unfortunately, we're limited by what we see. And we see ourselves as alive, and we see stuff around us as...
And in the first lecture, we had galaxies, black holes. They were sort of inevitable at that first instant of time. And I'm just wondering, if life is inevitable at that first instant. Is there a connection to be made from the first lecture to the second lecture in this series?
Say yes, I think it's a very about. figure
Are
out.
we Calvinists, I guess?
Just trying to see if there's some connecting point between the two, or are they just in two separate boxes?
Well, I think the point is, I mean, if I understand Lowell's direction in this, is that he wants to get to understanding people and how people got here. And he's looking at it from a couple of different perspectives, including the chemicals perspective. The next one will be biological perspective, and there'll be a theological perspective, an anthropological perspective. So I think we're trying to look at the question from a lot of different ways, and we're just starting here.
The other thing is you couldn't have those chemicals unless you have exploding stars or star formation. Those chemicals don't come about until that happens. You only have hydrogen, helium, and some other elements in deep space. You've got to form a sun that has a furnace inside of it that creates all the other elements. So you don't have the chemistry until you have a star, and then it explodes and spokes it out all over. So there is a connection.
Another question in the back?
I was wondering, you mentioned that there might be like other ways that we don't understand. I was wondering if you had any specific examples in mind of those other ways, or whether that was just something that was really beyond the grasp of human imagination.
I guess the short answer is I really don't have any ideas. It is human imagination. I mean, that's what science is in effect. You take a look at an observable, and then you use your imagination to go beyond that observable. The trick with finding other life is that you have to find this initial observable, this initial thing you look at, which make you think that there might be something beyond it. And it's such a big leap. It's hard to figure out where to focus to begin with. So I think that's the issue. Or there could be no other life at all, and we could just be speculating a little bit. So I guess the short answer is no.
I'd like to thank Barry for his. Thank you all for coming, and please come back next time where we'll try to make a further connection between the creation of the universe, the origins of life, and Todd Jackman will be talking on November 8th on where animals come from, the beginning of animals. And, so hope to see you all back.